Good afternoon. You've joined us on Likeable Science, broadcasting live here from the ThinkTech studios in Pioneer Plaza. I'm your host, Ethan Allen, and Likeable Science is all about why you ought to pay attention to science, why you ought not to be scared of science, why you ought to love science. And we hope to show in this show why science is fun, interesting, entertaining, and educational. To help me on that task today, I had Dr. Carol Diebel, who is the Vice President for Public Programs at the Bernice Pauhi Bishop Museum. Did I say that right? Pauhi?
Pauhi, yes.
Pauhi, I never get that one right. Carol has had a fascinating career in wide-ranging education work experience. She got her bachelor's in biology from Humboldt State University and did a doctorate in biological oceanography from MIT Woods Hole Institute of Oceanographic Institution, I guess. She then spent about 20 years of her life doing all sorts of fascinating research projects, everything from studying how lobsters smell, I think you were in Florida at that point,
Yes.
To determining the magnetic sense of trout when she was down in New Zealand. She's also spent many years working in museums, being first a curator of marine biology at the Auckland War Memorial Museum, then the director of natural environment in the Museum of New Zealand, and most recently, before she came here, she was the director of the University of Alaska Museum of the North in Fairbanks. So she has bounced around the world widely and done all kinds of interesting stuff. So it's great. So how did you get started down this rather strange and circuitous path?
In a strange and circuitous way, probably. When I was about 9 years old, my family made its first trip. I lived in California. We made the first trip up and over the coastal hills to the ocean. And from the moment I saw the ocean, I knew that I wanted to live by it. And I figured if I was going to live by it, a really good way was to work on the ocean. And so I did that, and I've always been fascinated with animals and water and things like that. And so my undergraduate degree was in biology, but emphasizing the marine environment. And my PhD was in biological oceanography. And then I also did a lot of things like look in old bookstores and find things like this illustration that I have here. I don't know if you can see it. So that's a submarine fantasy from a book by William Beebe, who was one of the first people to go down in a submersible. And so from the moment I saw that, I thought, I want to go down in one of those. And that's what started me.
Excellent. And I gather you got your wish. You actually did go down a number of times. You've been in some submersibles,
yes.
I gather.
Yes.
That must be very both exciting, but also, I would think, a little bit frightening.
Yes. I think that if they would have plugged me up the way they do astronauts when they go into space, if they would have plugged me up the same way, they probably wouldn't have let me take my first dive. But from the moment I was in the water with that first dive, everything changed. And it's just like the submarine fantasy.
Uh-huh. There is something very compelling about the undersea environment that we so rarely see, but it's such a sort of beautiful, different world down there.
It really is. Yeah. There's so much we can learn.
Indeed, and you spent some time initially studying really one of the key groups in the whole ocean system, the plankton, right? Yes,
I worked on plankton that lived with each other, and nobody quite understood. So I worked on these shrimp-like animals called hyperid amphipods that live in the middle of the water. So they never encounter any kind of land or a boundary, but they spend some part or all of their lives trying to find jelly animals like jellyfish and a particular jelly animal called salps. And so the animal that I studied, you can see here, one of them I studied was this little guy called Phronema, and it has these big, huge, beautiful eyes, and that's what got me interested in him. And so I started to study the sensory biology of these animals. What you see, the barrel, that's the salp. And if we go on, that's what the salp used to look like. And then the Phronema ate all the insides out, carved it out, has a special tannin that tanned it. And then it lives inside it, has its babies, and goes on, and the circle continues.
Huh.
So I wanted to know, how did they find these salps in the middle of the water column?
Yeah. And what's the answer?
Well, it's really interesting. It's one of those things where you think that's what you're going to do.
Right.
And what you end up doing is things like this. These are picture shadowgraphs that I took of salps. This is a salp here. And we wanted to know what kind of a smell did they leave behind in the water, and what did it look like. And so, you know, I had to work with fluid dynamics people at MIT, and we figured out this technique, which we did out at sea in a little aquarium that we just built out of leftover stuff. And we did something called shadowgraph, and we captured the trails that the animals left behind. And from that, you then had to use all of your math and your chemistry and your physics to understand the trails. How long did they last in the water? And were there things in there that things like phronema could find, and then follow on up and find the hosts? So that's just one little aspect of it. And so what I essentially did is find out that the animals did have things equivalent to noses, that they were very good at smelling, that they could differentiate the hosts they liked from the hosts they didn't like, and that they could do it at a scale that not even we could comprehend.
Amazing, amazing. So those big, beautiful eyes that you commented on aren't really for finding their prey at all.
No, and that's what everybody thought they were for.
Right.
And so, you know, that's what I started to study a little bit initially, was the eyes. But in actuality, they can smell far before they can see. So they'll come up on an animal, and then they'll use their eyes to determine, yep, it's definitely the right one.
Ah, okay.
But they can do that on smell alone as well.
Chemistry drives so much in the animal world.
Oh, yeah.
Even among humans, of course, we are much more reliant on our sense of smell than we ever sort of recognize or realize, but much more powerful in a variety of other animals. I used to study the reproduction in garter snakes. That's entirely driven by sex pheromones.
That's right.
You can get males to actively court a paper towel that has the female sex pheromone on it, just as vigorously as they'll court a female. So it's powerful stuff, chemistry. You know?
Yes, it is.
Excellent. So then, what sort of moved you away from plankton, and were you on to lobsters next? Was that where you?
From that, I got a real interest in the sensory biology of it, because part of for my thesis, I had to find what was sensory on these animals. And so that required me doing a lot of microscopy work and all kinds of things in order to figure that out. And so I got really interested in the sensory biology. And that kind of naturally led into maybe doing some recordings of the sensory cells. And so my postdocs after that were learning how to do neurobiology. So I learned how to record from, like for the lobster example, I learned how to record from single cells in the brain of a lobster. And what we were trying to do was map the brain to figure out which parts had to do with smell.
Right, because lobster is one of those wonderful invertebrates that has a relatively small number of cells in its brain, a relatively small number of big cells in its brain.
It has, yeah.
That are essentially the same cell occurs in every animal, and you can find that same cell repeatedly in animals and eventually track down its function if you
That's
play
right,
enough
because
games.
what people found was that actually it was a lot easier to study these cell types in lobsters than it was in humans.
Yes.
And yet we could use the information we found to help us direct what we then needed to study in humans.
Right, and there have been a number of course classic invertebrate preps for neurobiology.
Right.
And, Aplysia snail being, of course, big one, but lobsters have been widely used too. That's great.
That's right.
And then you went on to study a much more obscure sense a little bit later, right? That is the magnetic sense in trout, which now magnetic sense, of course, we have no real analog for, right?
Right, and so for magnetic sense, what happened was that I developed all these skills for tracing individual cells, looking at their morphology, trying to figure out where they were. And the person that I did the work with, Dr. Michael Walker at Auckland University, had been doing the behavior of fish and had been able to see that they could respond and be trained to the magnetic fields. but there was no idea where the receptor was. And so he had started recording from some of the larger nerves and found what he thought might be a signal going through when he changed magnetic fields. And so that's when I came, and then I started to trace that nerve throughout the fish and try and find the cells or the sense organ that was responsible for magnetic sense.
Interesting, interesting. Almost, I went through almost the reverse experience in my work with fish, in that these fish, these were African cichlid, had been examined, and the retinas had these well-organized sets of cones, paired cones around a single central cone, very strongly suggesting, of course, that they have color vision. And then I ended up doing the behavioral work with the fish to help demonstrate they actually are making decisions based on color. So,
right,
right.
That's right.
Just almost a backwards process there. Interesting how science does not follow a single model, right?
No,
different ways.
It's amazing. But you can, like in the case of the lobster, even though if you looked at it, it looked completely different. If you looked at it from a systems approach, it was very similar.
Right.
So you had to ignore the fact that the cells looked different, they were in different locations, they were arranged different ways, but functionally they did very similar things.
Right, right. The invertebrates all face the same constraints in life and basically have the same jobs to do fundamentally, and they've sort of organized their nervous systems rather differently to do those.
And if you're in water, it's a little different than if you're in air.
Right, exactly. So that's intriguing. So you did all this great research, which I well know research is a lot of fun to do. It's challenging, exciting, and all. So what was it then that drove you to shift and get away from research and start moving more into the line of education?
I think that for me, I'd been doing research for 20 years, 25 years almost, and on something called soft science, which meant I had to essentially find my own salary through grants all of the time. And so you're in kind of a 3-year cycle for doing that. So you get 1 year when you are writing all the grants like mad because you have about a 1 out of 10 chance of getting them. So you're writing a lot of grants, and then if you get the grant, then you actually have about a year or two to start to do it, but then by the 3rd year you're having to write the next grant in order to get the money to do the next step. And so about 25 years of this, I was getting a little bit tired, and my family kept saying, when are you going to get a real job? And the opportunity came up for a job in a museum. I hadn't been that aware that there were jobs in museums, and I had a lot of background in taxonomy because I'd done that as I'd worked my way through school. A lot of my jobs had been as an invertebrate taxonomist, and so I had some of the skills they needed for the curatorial area. And so I transitioned over in about my 25th year to a job with an actual salary that was dependable. And so that was pretty nice. Also, universities just didn't seem to want me. You know, I just didn't fit their, I'm a generalist much more than a specialist. And at the time that they were hiring, and probably still now, they look far more for specialists than they do for generalists.
I hear you. I went through that same thing when I got my degree. I was very interested in whole animal behavior of large living actual animals, and all the trends were to look for at that point single cells or single receptor types or single molecules that were of interest, and that's where all the funding was. That's where universities were hiring for, and yes, that's kept me away from that particular track.
That's right. But you know, it's like kind of a blessing in disguise, because you know you're challenged all the time to find something to do, and so from that probably suits me. I like the learning of new things all the time.
Indeed, it's always exciting to see new things, learn. I just posted on Facebook that I ran into a little piece that points out there are fish-eating spiders on every continent except Antarctica. I had no idea there were any fish-eating spiders. And I mean, I have some fair background in biology, but I didn't know spiders ate fish. You know, so that was a new thing for me.
Yeah, that's pretty neat.
So anyhow, in the museum world, putting together an exhibit is a very complex process because it takes above and beyond this sort of the research skills. There's a whole bunch of other aspects of it. And I thought maybe you could sort of walk us through a little bit of that process, perhaps.
Okay, what I thought I'd do is give you some examples of different kinds of exhibits.
Sure.
And just kind of show you how the process is somewhat different. So the first exhibit, one of the first exhibits I worked on was called DNA, the X Factor. And it was celebrating the discovery of the structure of DNA. And I was living in New Zealand at the time, and one of the scientists was a New Zealander by birth. And so we were celebrating that and the anniversary of it. And so I worked on this, and this is an example of something where that's not my area of expertise at all.
Right.
But I had to do an entire exhibit on DNA. And I learned a huge amount by doing that.
Isn't that the way it goes in museums? Yep. My first job in a museum, I went to work out of a postdoctoral fellowship, having been a behavioral biologist for many years. The first project that Museum of Science and Industry put me on was fusion energy.
Yeah, exactly. Just like, okay. So I learned how to, I created a 9-minute movie, and I had learned how to write a script, and I learned how to block things out. I learned how to help create interactives. I learned how to program things. I learned so much by doing that. But if I hadn't been a scientist beforehand, learning how to do all those things as well in order to do my work, it wouldn't have happened.
Right.
You know, I really learned about imagery and things like that when I was doing my research. So it helped quite a bit.
Yeah, absolutely.
Yeah. One of the second things I did was called Weird and Wonderful, which was a children's gallery of discovery at the Auckland Museum. And what this was is a case of an existing gallery that needed to be refreshed. So what I did on this was work with an entire team of people, of course, and I was the content expert. And so, you know, I worked with the project manager to work on this. So for a gallery design of this, here's an idea of some of the steps that you might go through for a gallery. So first you have to decide on a concept. What are you going to do? What's the and this was a children's gallery, so what kind of learning do we want to have in it?
Right. What are the messages?
What the messages are, or the way we want them to interact with the gallery. Then of course we defined the audience, so we knew historically what groups went there, you know, school groups, family groups. So you wouldn't design this gallery for a university student.
Right.
So, you know, that's all goes with helping you decide the way you're going to present information.
Sure.
And then once you decide the concept, then you go and you work with the designer and you start to look at all the different ways that you might design this gallery. And so this, for us, the decision was that we wanted the kids to have a sense of self-discovery. So it was a completely packed gallery, but all kinds of nooks and crannies and places for the kids to go. It was not a lot of information. It was the age of computers, so they could always look in computers. So it wasn't, labels weren't it. It was discovery, but they could then go to a computer station and look up more if they wanted. So there was that. There were quiet places that the parents could sit, interact with their kids. There were places to put parents who came in with 2-year-olds. We had spaces so that their hands were free. All those kinds of things you kind of think of. So, you know, the elements we did were nature, because this was a nature gallery. So that's the kind of thing. Just to show you, just some of the things that came out of all of our work was kids love to perch. They love to view from the top down. So we created a perch in this gallery, and the kids could climb up it, and here's some pictures of it kind of looking down into it. And if you look in the gallery, you can see how just completely full of collections, objects, and stuff that there is in it, and all kinds of ways to inform it. Here's another case where we broke the rules. People said we couldn't have collections and alcohol in the rooms, which is a real problem. So we worked and figured out a solution that we could put them in, and we used animals that weren't totally unique. They were duplicates that could be used for education purposes. And so we put them in the jars, and we did things like we made these little labels for them so they could see a picture of what that animal really looked like, and each of the labels had a fun fact on the back. And what happened is the schools would come in and start to use these, and the kids would have to find information on something in it. And we also did a few things you see here, like there's Bart Simpson. And so, we put him in a jar, Bart Simpson-I, and, if they could find him, and it forced them to stop and look in the gallery itself.
Sure.
So that's an example of one. And this gallery won an award. It won a design for a best public space. And what I loved about it is there's a write-up that they did for it, and they encapsulated better than us what we were trying to get across.
That's wonderful. In
that gallery space. So I've stolen what they wrote many times to describe what we were really trying to get at in that gallery. So that took about 2 years to do with that particular one. This is just as you're doing things like this, you start to get art in with your science.
Sure.
So, there's some things like that. And there's, one where all of the snails were put in a big spiral like snails are. This is another one. This is where it was an opportunistic experience where a huge squid was caught in Antarctica by fishermen. And they brought it on, so this is a picture of it while it was still alive along the side of the ship. These were Patagonian toothfish fishermen, and this was caught eating one of the fish that was on their line. And it was probably down about 3,000 feet when they caught it. So it's a classic case where it was brought up, they froze it in a big fish container and brought it back to us, and it took a year for all of the politics and the red tape to really actually release it to the museum so that we could do something with it. And so this is just, it had all the politicians wanted to come down and get their pictures taken and all that other stuff.
The dead squid, of course.
Yeah. So here you are doing an exhibition, but I'll tell you, we had to write a deed of gift in order for this to go. It had to go through lawyers, it had to go through the government, it had to go through all the ministries, and because this was in New Zealand, in order for us to then take something to go forward with it. And here's another case with this exhibit where to thaw it out, we needed a special tank made. We had to have an engineer work with us because of the load of that tank on the floor. We couldn't exceed it. And so we had to work with an engineer in order to create a tank that would hold that volume of water, not be too heavy, not cause problems, and that we could still use to work to thaw it out. So there's another example. Here's another where here we are, the colossal squid is in that blue tarp somewhere, completely frozen. It's been frozen for a year. You can see our beautiful tank that we built underneath it. And now we're going to thaw it out in the public with absolutely no idea what we're going to find, what we're going to see, and what's going to happen, how long it's going to take. And we streamed this on the internet. It was one of the first streamings on an internet. And we just put it up in black and white with no sound because we had very little money. And we just put it up to see. And it crashed the internet.
Because so many people.
Because so many people were doing it. Crashed a node in San Francisco alone just because they couldn't do it. And so all of a sudden, the money appeared for us to go in color and to have sound. And so we had it up and did this over the course of a few days while we thawed it out. That's the end result. So when we thawed out, it was in pretty good shape. It had shrunk significantly from being frozen for so long and water being removed from the cells and shrinking. We didn't know. No one had ever caught a whole one of these before. And we wanted to know about the eyes. And we didn't know if there would be eyes. So we had people who flew down from Sweden just in case the eyes were there. And there was one intact eye that turned out to be the largest eye in the animal kingdom.
Huh.
So, and they took it back and worked on it. So that's the thing. Here's another thing, like funny little details. Okay, these are not giant squid. These are colossal squid. One of the ways you can tell the difference are these little hooks, and they swivel.
Huh.
Giant squid don't have hooks that swivel. They have these teeth-like things, but they don't have these.
I see.
So there's a detail.
Right.
And then because this was so popular, we turned it into an exhibition.
Okay.
Okay, so there we go.
Right.
And then here's like a board, a storyboard that we've started to go with. What are we going to do in the exhibit? Scientists don't know anything about this.
Right.
People get very nervous when you put on an exhibition where nobody really knows
Sure.
what's going on. I feel we just could turn it around and say, we don't know, what do you think?
Right. Right.
And that's
Make it participatory.
make it participatory,
Right,
and
right.
that's what we did. And when we opened the exhibition, we had lines for months out the door to see it. We had to do crowd control for it. So yeah, and this just shows you the kind of detailing that we do.
Mm-hmm.
You know, here are steps in a video that we did on the squid. It was a 3D experimental new thing. So we had a little teeny theaterette, and we actually did a 3D of a colossal squid and capturing food and doing things like that. So these are all of the story parts of it. And then here's some of the design planning for the exhibition. So you can see the amount of work and detail that goes into doing something like this. It was only supposed to be up for about 3 months, and it's still there.
Wow.
I don't know, it's 5 years or more now. So yeah,
Yeah. Good exhibits will have a habit of sticking around.
that's right. So, and I don't know if you want to go into, do you want me to talk about the whales now? Or
I tell you what, I think we need to take a quick break
Right.
here, but we'll do that when we come back. So you're watching Likeable Science with me. Your host, Ethan Allen. Today in the studio is Dr. Carol Diebel from the Bishop Museum, and we're talking about her experiences in doing ocean research and also museum work. We'll be right back.
Hello, I'm Martin Despang, and I'm the host together with the one and only Ali Amashta, and our show is called Urban Transcendence. And Urban Transcendence is about identifying where we have a unique situation of a vibrant city in one of the most beautiful natural environments. So how these two can coincide, sometimes conflict, how they could find reciprocity in the 21st century is what we're excited about. And we're planning on bringing in a diverse body of guests, both from the arts and the science and the established and the wise and the emerging generation. So hope you will join us. We'll always be on Thursdays from 1 to 2 p.m.
And you're back on Likeable Science. I'm your host, Ethan Allen. With me today here in the ThinkTech studios is Dr. Carol Diebel. We're talking about her experiences in ocean research and also in museum work, typically around ocean themes. So she was telling us before about some wonderful exhibition development that she did on well, several exhibits, but the most recent there about the giant squid and all the whys, wherefores, and different ways that exhibit came about. But you've also now got a much more current exhibit, right, about whales that you're doing, which strikes me as a big topic. And it was one of the questions, when you get a big topic like whales, how do you decide sort of what aspect to do, what to talk about, what not to talk about? You know?
well, I think it's really interesting because when I first started at the National Museum of New Zealand, the whales exhibition had been kind of started. There'd been some write-up of it, and they were starting to go. And they were taking what I would call a very traditional, educational view of it. You know, the classic things, how thick is whale blubber? How does it keep you warm? You know, how do they sieve their food if they have baleen? You know, where do they live? What do they do that way? And when I came, I was much more interested in exploring a part of whales that only that museum could do, and that the New Zealand scientists were working on. And so we had the most amazing collection of whale specimens, whale bones. We had an amazing curator, or actually technically he was a collection manager, Anton van Helden, who really was fantastic about relaying information in a public-friendly way. And from where I'd come with my research background, I knew that there was a tremendous amount of research that was going on in the whale research community that had not come out to the public yet. And so I thought, why don't we put all of these together to do an exhibition from the science side? But in New Zealand, Te Papa, or the National Museum, is a bicultural museum with the indigenous people, the Māori.
Right.
And so what was really important to me as well was to include that component of it, the cultural component of it. And in order to do that, you really have to slow the whole process down, because there needs to be a lot of dialogue
Right.
with all of the different Māori iwi or Māori groups in order to see if they were interested in working with us, if they wanted to, what stories did they want told, and how would we put this together into the exhibition. Because a lot of people at that time didn't feel that you could mix science with culture.
Right.
And I feel that they strengthen one another, didn't weaken one another.
Exactly. But,
Right.
but it's not always exactly an easy marriage, because they're quite often quite different in terms of the approaches to how you deal with information, for instance, and how information gets shared.
Exactly. I mean, a really classic example is taxonomy of whales. If you look at the way whales are related to one another from a scientific point of view, you use all the morphological characteristics and you use the biology of the animal in order to do it. From the Māori perspective, you use their place within the environment. And so they're not necessarily connected as much to other animals as they are to the whale equivalent in a forest, which might be a giant redwood. Or in New Zealand, it might be a giant kauri tree. But what that does is affect that environment. And those are equally fascinating ways to look at where does a whale sit in the world. And so that's the kind of thing that we looked at and we started to explore. And so if I take you through quickly the whales exhibition, I just want you to see that the planning for it started in 2004. The exhibition didn't open until 2007.
Wow.
So that was 3 years. And so we had to get a concept, we got the funding, and we had to get the museum to commit. But also there are a lot of things, like I talked about with the colossal squid, that we had to do with the whale. What precipitated the interest in doing this exhibition was there was a stranding of 17 sperm whales on a beach up on the northwest part of New Zealand. And so we got the skeleton of an 18-meter or 28-foot or so sperm whale. And what we wanted to do was and especially our collection manager, Anton, was totally committed to this, that we wanted to articulate the skeleton the way a whale actually sits in the water. If you look in the old museums and you walk in and look at the whale skeletons, they have put it together like it's a great big huge water horse or something, not the way it actually sits in the water. And so what we were interested in doing was, how does it really sit in the water column? How do we make it so that it's not obviously articulated with wires and straps and things like that? And since we were going to tour this, how do we attach it in crates so that we only have to drill one hole for articulation, one hole for the crate? And that took us over 2 years to develop this new method of articulation. But it has allowed these to tour. And it opened in 2007, and this exhibition is still touring in the U.S. right now.
Wow.
It's at the Fernbank Museum in Atlanta right now, and it goes to Denver after that.
Well, that's great. Yeah.
Yeah. So that's the kind of thing.
It's a testament to the quality of the product, right?
That's right.
If people keep wanting to see it, and yeah, excellent.
So here's kind of the first steps that we did. We did a concept so that when we got the elders together, the Māori elders, we could go over that idea with them. What did they think of it? How did they see it? And then we did and we did the same thing with the scientists. And then we refined all of our concepts according to what they said or their suggestions were. We then did something a little bit different where Anton and I traveled to whale scientists, and others in the team traveled to the different Māori elders, and we sat with them and asked them, talk to us about your story or the research that you're doing. And we did that around the world and came back with what we thought were the most compelling, interesting stories that nobody had told as yet, or else they were old stories that had a whole new twist on them as a result of the research. So we had to come up with the big idea.
Right.
We also came up with the bicultural objective.
Okay.
And so once we'd done that, we broke it into segments, because in an exhibition this is about anywhere from 7 to 10,000 square feet. So you break it up into segments. And we went through and we talked about New Zealand's in the South Pacific. So these were whales that were around New Zealand, which are whales that a lot of the rest of the world doesn't see very much, beaked whales in particular. So again, it was a unique aspect to it. We talked about whale people. So these are the Māori communities, and also some of their past from Polynesia. We had something we called a whale lab, where we put a lot of the science that was going on within it. And then we had another one which was actually a combination of culture and science, which are whale strandings.
Right.
And that's where both sides meet on a regular basis, at least in New Zealand, they do. So that's the kind of segments. So the South Pacifics, when you enter, we want people to know where they were and what it looks like. The whale people is people telling their different stories. One is in particular, which some people might know the film Whale Rider.
Sure.
And so we talked about whale riders in New Zealand, and so there were the stories of different groups that did that. So here, and we constructed this from an old prop that was used for the Whale Rider film. So we created a theaterette out of one of their old, one of the whales they had used in some of their stranding shots. So it had that. There's the stranding. And so this is the articulation of the 18-meter or 28-foot or so male sperm whale. The females behind there, you can't see her too well. But it's suspended it's unusual in that for the most part it's suspended from the top, but it also has anchors along the bottom to hold it. And we did that because if it was going to go to different museums in the world, they had different capacities within their exhibition halls.
Sure.
So it allows us to adjust according to that. This is one of the Māori elders in the area where the sperm whales were, and he tells the story of how the sperm whale is a part of their culture and their lives. Okay. So you get that. Yeah. The opening of the exhibition was amazing. We had 11 different iwi, or 11 different groups of Māori who participated in the exhibition. Each one of them sent their elder. Each elder blessed the exhibition. And so we had whole ceremonies around them doing that. These are some of the lessons that we learned from putting culture and science together. Probably the first one is the most important. You have to be patient.
Yes, indeed.
You have to listen, and then you have to listen again. And that the most important thing is to learn a dialogue or a vocabulary that both of you understand, both of you agree on. And that's when you start to really move along and make some progress.
It's intriguing to hear this. We face very much the same challenges and issues and have come to the same realizations in our work in more general education throughout the Pacific Islands and in Prel, where I work.
Oh, that's interesting.
And we absolutely have to go and find where the Yapese or Truckese culture or whatever it is you're working with, where they stand, what do they want to do, what do they already know, what's their worldview. You can't just sort of walk in and say, this is science and you must do it this way. That's not going to work.
No, it's not. And you also can end up like Margaret Mead, where they could create this whole shadow thing of what they do because they don't really trust you enough to tell you what's going on. Mm-hmm. And that was a part of the process that we had to get through. They had to trust us enough to give us their best people, mm-hmm. so that the best stories could be told.
Right.
But you have to earn that trust.
Absolutely.
And you know, that made this exhibit probably take an extra year.
Yeah.
But I really believe it was totally not only worth it, we had to do that.
Right. A really vital part. And,
sure.
And building that relationship is key to any of these good collaboration.
It's the same thing with the scientists.
Right.
They had to trust us to present their information.
Right.
In a good way. We did not do it at all the way they wanted us to. Mm-hmm. But they were really happy with what we had done once they saw it. Right. I think that's the difference with museums.
Right. Most scientists, of course, are not particularly well trained as communicators, other than perhaps other scientists working within their same relatively narrow field.
Right. And teaching a class is not presenting it in a museum. You can't. Right. You can't present it that way. People just aren't going to engage. And so that's what we do. And that's a real particular rare skill set that a lot of museum people have, is that ability to understand how to explain something in a way that everybody can understand.
Exactly. I recall working on an exhibit for atherosclerosis many years ago, and my collaborator was a very eminent retired heart guy, and wrote these wonderful 3-page long, totally dense, detailed text explanations of what these exhibits were that no visitor was ever going to read.
That's right. Yes.
And then it was my job to take his 3 pages and turn it to half a dozen pithy short sentences. So that was always a fun challenge.
Yeah. In the whales exhibit, one in particular that I'm really proud of was we worked with a research scientist who attached data tags to the backs of sperm whales just before they would go down for a dive. Mm-hmm. So they would attach them to the back, the whale would go down, for its dive. Eventually it would come up, and when it came up, the data logger would pop off and they would collect it, and then they would analyze the data. So I'm sitting in the lab with this guy with headphones on, listening to the whale as it's doing its dive. And you can hear the heartbeat. You can hear their flukes move. You can hear them as they move breath around. And, I'm going, wait a minute, they're in the water. How are they breathing, moving breath around? And one of the things they discovered from all of this was the way they do their sonic pinging is they have air, which they recycle through different cavities, which they then use to send out sound waves in order to look for prey. And so I said to him, how do you show people what this dive looks like? And he showed me, as a classic scientist would, and as I would have had I still been doing this, a series of graphs that showed the different information. And I said, no, but how do you really show them?
Right.
And so what we did was we worked with him. We took the 3-hour recording. We got it down to 3 minutes.
Right.
And we did a 3D visualization of that whale diving down, doing the things that you could tell from the data logger that they were doing. And we showed the way a whale sends out the sound in order to ping for, and we show the way they get the sound back and how they hear it, which was totally new science, which a lot of people still don't even know, and it's how many years later. But that's what we showed in it. And people walked out of there from doing the surveys totally understanding what they were doing. And not only that, then the researcher had that, which they could take and show when they gave talks.
Right.
So we created all of this information, which all of these whale researchers could then take and use when they gave talks.
Oh, that's wonderful. It's a nice, again, a nice bridging between the fields,
Yes.
between the research and education areas.
Yeah, it's great.
Super. That's wonderful. And so you touched on a topic there that I wanted to get a little bit into, the value of sort of technology in all of this. We're here on ThinkTech Hawaii, and it's one of the big thrusts here is how is technology being of service to us? And you spoke of those data loggers and other technologies that clearly had to be employed to get this out. So do you see technology as now being helpful to your museum education experiences, or do you see it as, in some sense, limiting them?
I never see it as limiting them. For me, technology really stimulates constant innovation and constant new ideas. So, someone comes out with a piece of technology and we look at it in a way that maybe it wasn't intended, but we see we could use it. And so that's what we do. We use it that way. It totally changes classic science research. It also changes the way we can do interactives and innovate within the museum as well. You just can't uncouple them. The only reason that they ever get uncoupled is money.
Okay. Money will drive a lot of...
Money will drive it. If you want to do the latest technology, you're going to pay significantly more for doing that, and also spend significantly more time making it work within the context. And so it can be quite frustrating because everybody wants to be what they call cutting edge.
Right.
But really, a lot of times what you need is tried and true technology, which we might use in maybe a little different, more creative way.
Right.
Which we think will be more sustainable on the floor or for people in a more budget-friendly way.
Sure. Be more rugged, more reliable, stand up to...
That's right. Honestly, there's no reason to overkill with things too. If a simple flip board will work, that's what we'll use.
Right.
If we want an interactive 3D digital holodeck type thing, then that's what we'll use if we can get the money for it.
Right, yeah, exactly. It's got to be a nice match there between what message you're trying to convey and the sort of the medium or the technology that you're using to convey it. But my point had been that some people worry that technology somehow substitutes for the real things, the real experiences, the real objects in museums. But it sounds like you don't ever want to go that path.
Never.
Right.
But the thing is, what it can do is show you things about the object or that animal that, because we don't have it living, it won't do for you.
Right, you can go inside.
We can go inside and do it. A classic case would be an anglerfish. I worked on really early work on visualizing a 3D of an anglerfish because I'd collected them. I actually did have videos of them because we'd collected them in the submersible, brought them on board ship, and we'd done videos of their swimming and behavior, which, people hadn't really seen or done much of. But then, we want to know, well, what happens if you feed it? How does it use that little glowing globe, and how does it really move it around? And sometimes you need to go, so to do it by 3D, we could look at that. Also, by doing it by 3D, we could understand far better the way they use their fins for moving around, because you can then apply the physics to it and other kinds of things. So, it's a great collaborative relationship with technology.
Well, wonderful. This has been so interesting to hear that this wonderful blending of science, research, education, technology, and how they all sort of mutually reinforce each other. This has been a wonderful conversation, and I'm only sorry that it has to end now because we are out of time. You've been with us on Likeable Science. My guest today has been Dr. Carol Diebel from the Bishop Museum, and she's been telling us about her wonderful experiences in ocean science and all of the wonderful education and research that she's done over the years. And we hope you'll come back next week and join us again. Thank you very much, Carol.
Thank you.
We'll see you next week.